The next speaker is Pascal MILESI, and he's going to talk about genomic data provides new insights on the demographic history, local adaptation pattern, and genetic basis of quantitative traits.
Hello, everyone. First, I would like to thank the organizer for offering me the opportunity to present our work about the evolutionary history and local adaptation in Norway spruce. Ultimately, evolution can be defined as simply a change in allele frequency across time. And there is 4 main forces that can explain such a change: the mutation, the natural selection, the migration, and the genetic drift. And the interplay between these forces will shape the biodiversity. Among them, natural selection has a peculiar place. Of course, it is because Darwin described this process, but it is mainly because it explains why the phenotypes so often match their environment. And it is particularly true for quantitative traits that, for instance, change according to environmental gradient. For a lot of tree species, for instance, the height of the trees decreases with the latitude or with the altitude. And if we consider a plant life cycle, most of the life history traits, most of the traits that matter the most for the fitness are quantitative traits. And in some way, fitness itself can be perceived as a quantitative trait. So it's a matter of importance to understand the genetic basis of these traits. However, as we are going to see, it's not easy to determine the genetic basis of these traits and to infer the role of local adaptation in their determinism, in their variation. First, the selective pressure are often multiple and correlated because the adaptation takes place along environmental gradients. For instance, in these 2 graphs, you can see the mean temperature as a function of day duration or mean precipitation at sampling site in the study that I'm going to present to you. And as you can see, all are strongly correlated. And the colors reflect the latitude. Okay, so as the trees were collected along a latitudinal gradient, all the climatic variables that varies according to the latitudes are autocorrelated. And this is only a biotic factor. But of course, the environment is also made of biotic factors, right? For instance, pathogens are not evenly distributed in the environment, and we know that pathogens can exert a very strong selective pressure on the individuals. So you have to also keep in mind that in the study that I'm going to present, we have no idea about this, right? Okay, second problem. The quantitative traits have also often a very complex determinism. There is a lot of genes that are involved in their controls, but there is also non-genetic factor. For instance, epigenetic factors that are going to modify the phenotype, but also, of course, the environment will affect the phenotype. And what we are going to measure is this, right? But what it is transmitted to the next generation is that. So it means that at some point we have to be able to disentangle the effect of the environment and the effect of the genotype on the phenotype. And last but not least, the phenotype of the individual, when you are going to measure them today, it includes all the evolutionary history of these individuals. So it includes the effect of natural selection, but also the effect of random forces like genetic drift. For instance, in that gray environment, you have different populations with that exchange genes, a lot of gene flow, and for some reason, the environment can be fragmented, so the gene flow will be reduced between the populations, and we can imagine that in a very extreme case that the populations are not connected anymore. So all the gene flow is within population and no more between population, just the individuals within population exchange genes. The mutation being a random process, different mutations can be fixed in the different populations. Of course, those mutations can affect the phenotype, and it happens that the phenotypes varies from one population to another just because of random processes. So the sample match between different population, different phenotype does not have to be linked to natural selection. Of course, the environment also often varies in different populations. So natural selection plays also a role. But what I want to stress is that the phenotype is the result of both the effect of drift, and the effect of natural selection. And again, we will have to be able to disentangle the effect of both if we want to investigate the local adaptation. We have seen this morning that there is a way to assess, and there is experimental design that allowed us to separate and to disentangle all these forces, and among them, of course, common garden experiment. And the idea of our study was to use the extensive breeding program for Norway spruce that is ongoing in Sweden as a massive common garden experiment. For that breeding program, that breeding program includes trees that had an evolutionary history in the whole ranges of the Norway spruce, right? So it means that all the trees that had an evolutionary history in different environments were planted in the same and single environment. And this will allow us to disentangle first the effect of the environment and the effect of the genetic on the phenotype. The basic idea is that if there is differences in phenotype, it is due to the genotype and not due to the environment. It's a rough opinion, it's a rough idea, but just to be schematic, right? So then we sequences 1,500 of those trees so that we get access to the genotype. And this allowed us to investigate the demographic history, so that we'll be able to take into account the demographic history, to correct for the demographic history in our search for local adaptation. We also got access, thanks to the Forest Research Institute of Sweden, to the phenotype of the trees. We got access to the height, the diameter, and the bud burst. And in the last step, it means that we would be able to identify the genetic basis of those traits while correcting for demographic history for population structure. But first things first, demographic history of Norway spruce. Based on roughly 500,000 SNPs that are neutral, putatively neutral, and linked, we infer, on that graph you have the time which is running like this, we infer the different splits that occur between the main species of that phylogeny, and we infer also the change in effective size. So for instance, 23 million years ago, Picea morica, which is found here in Serbia, diverged from the common ancestor of Picea obovata, Picea abies. Picea obovata is found mainly in Siberia, and Picea abies, Western Europe, right? And about a given number of million years ago, there was a split between the main domain of Picea abies within the species. So we were able to infer the timing of the split, and we were able to infer the change in effective population size. For instance, we estimated that the ancestral population was about 700,000 trees, while for Picea morica today, we estimate the population effective size being 78 trees. Okay, so this can help to synchronize and to take decision about the effort we can have in a conservation perspective, for instance. This kind of methodology can help you to decide, okay, there is a need for that species we should take care because the populations are very low, because the genetic diversity is very low, etc., etc. And for Picea abies, we are about 7,000 trees within each of the main domain. So that split led to the main population structure in Picea abies. The Carpathian mountain, the Alpine mountain, and the Phenoscandia represent the main refugia during the glacial maximum, right? So when the ice covers the lands, the population tends to be isolated, and then they tend to diverge. But of course, when the ice melts, the population tends to expand, so they enter in secondary contact, they start to hybridize, and this gives rise to the population structure that we have currently in Picea abies. This principal components analysis is based only on the 500,000 markers I showed you, and you can see that the Carpathian, the Alpine, and the Phenoscandia domain are the angle of this triangle. They are the ancestral refugia, and that, for instance, Central Europe is the result of the recent admixture between Carpathian and Alpine. Okay? So now that we have a clear view about the demographic history and the population structure, we can look at the pattern of local adaptation. The fact that now we are able to identify where the trees have had their evolutionary history, it means that we have been able to go back to the ancestral environment to look at the variation of that environment. So here you have a principal component analysis based on climatic variable recorded at the location of the ancestral environment of the trees. So all the trees were grown in Sweden, but their history were in different places all over Europe, right? And you can see that there is exactly the same pattern of variation between the ancestral environment and the genetics. But this is globally expected. Then we look at the same principal component analysis, only based on the phenotypic data. So you can see barely nothing, you have to trust me, but we have, again, the same triangle, the same pattern as the Picea based only on the genotype and the environment. And this is more visible here, for instance. This is the correlation between the principal component for the phenotype, the principal component 1, with the correlation for the principal component 1 for the environment. And you can see that they are strongly correlated. There is a quantitative relationship between the change in phenotype and the change in the ancestral environment. So this highlights a very strong pattern of local adaptation. We were able to look at the value of the phenotype of the different phenotypic traits we have regarding the genetic clusters they belong. And despite the fact that all the trees were grown in Sweden, southern provenance performed much better than local provenances. So trees coming from the Carpathian, the Alpine, or Central Europe are bigger, are taller than trees that have their evolutionary history in Sweden. So this suggests that we can use, for instance, strategies as assisted migration to anticipate the change in environment. To use the idea that this population includes pre-adapted genes for the future environmental condition we will have in Sweden. Now for the bud burst, the pattern is a bit different because bud burst varies according to latitude and longitude, and in Western Europe it varies across along continentality. So we know about demographic history, we know about the variation of phenotype. What about the determinism of those traits? So we conducted an approach through GWAS. And this is for the bud burst, what we had. We detected a lot of SNPs that were involved in their control. And this is what we had for the diameter. I have to say that for that we did not correct for population structure. I did this because I wanted to stress the point that it's really important to take into account population structure. It's really important to deeply infer population structure. If you do not correct for the demographic history, you detect about 300,000 SNPs as being involved in the control of the diameter. Okay? So then that's the result when you control for population structure. And something that we can afford. We can work with that. So we show that for the bud burst, the genetic architecture is made of few genes of big effect, right? While for the growth traits, the genetic architecture is highly polygenetic. So it's lots of genes that are involved, all of them having a small effect on the trait. So then we look at the function involved, the function in which the genes were involved, etc., etc. So it allowed us to propose candidate genes that can serve in a breeding perspective, for instance. So to conclude, thanks to the genotype, we were able to infer the ancestral environment of the trees, where did they have their evolutionary history. We link the data from this ancestral environment with the genotype, the phenotype, to assess the pattern of local adaptation. We also link the genotype and the phenotype while correcting for population structure and demographic history to infer the genetic architecture of these traits. And all of this just to say that this kind of approach brings new insight for fundamental aspects, but also for more applied perspectives. Just an advertising, Mathieu is going to present his poster at the session this afternoon. It's about Poplar, and I was just able to come with this. With Mathieu, Poplar is popular. So do not hesitate to go and see him. I would like to thank all the persons involved in that work, starting by Martin, that was my postdoc supervisor at that time, and our funding. And thank you very much.
So I thank all the speakers to be precisely on time. We're not a minute behind schedule. So any question to Pascal?
Sorry, Pascal. Well, I had too many questions, but I'm a little bit surprised by one of the things you said when looking to the figures of the phenotypes, the diameter or the bars, and you said, well, there are differences among populations and domains, so this open room for, it makes attractive, like a system migration, like to be used. But what I see also is there are differences in the means, but there is a huge variance within population. So I really I don't see the need to put in material for elsewhere just based on that piece of information. You have really high variance within populations. So,
yeah.
Would you agree on that? Or like?
Yeah. One explanation of the high variance is that the amount, it was not splitted by population, but by genetic cluster of origin. So it's quite wide. So of course, we can imagine having more studies at a more fine scale, right? Then, so the localities, the true localities of the trees varied within genetic cluster that can explain this high variance in the response. The number of trees for each, of course, cluster varied a lot. But what I want to say is that that was just like height and diameter and bud burst. But there is a lot of traits, of course, that we have to look at, as for instance, resistance to pathogens or resistance to cold. That's a matter of importance in southern Sweden. Those trees are able to resist to cold also, for instance. And so far, we have no clue that there is more damages on trees coming from the Alpine or from the Carpathian region in southern Sweden. This is just for southern Sweden. I'm pretty sure that if we had the same study, but in the northern part of Sweden, the result will be completely different.
Hello, Ivan Scotti from IRAE, Avignon. Great talk, Pascal, thank you very much. I had a question about your demographic estimate. I know it is notoriously difficult to estimate precisely single parameters like effective sample size or effective population size, or times and so on and so forth. So I just wanted to ask you how precise your estimates are, or what the variance of the estimate is, and how you managed to get them very precise if you did.
So to be honest, that part of the work was not done by me, but by June. I will try to help you, and if you want to know more, I can orient you to June. So the software we used was FastSimCall, developed by Escoffier Laboratory. I can tell you that we considered a generation time of 50 years for the Norway spruce, a mutation rate of 10⁻⁹, if I'm right. We tested for different generation time, for instance, and of course, it changed the timing of the splits, but it did not change the relative position of the splits. And this is what we were mainly interested in, right? It was not, the point was not to deeply link this with what we know from geological records of environmental change across time, for instance. We just wanted to have a global overview about the main scenario that happens. Then, regarding the effective population size, depending on the estimates we have, of course, it's changed. But when you go from 700,000 to 78 for Picea morica, the story does not change more if instead of 78, it is 140, for instance. So again, our point was just to have a global idea. One other part of that paper that I did not present here is that in the records we get access, there was a lot of blank, because when the breeding program was started in the middle of the 20th century, of course, the information was lost for a lot of trees. So one of our goals was to infer from the genotype directly, to infer the genetic cluster of origin of the trees. So that was part of this demographic analysis. The point was to be able to say, okay, that trees, you don't know where they come from, but thanks to the genotype, we can tell you that it is a tree that came from the Alpine, it is a tree that came from the Carpathian. So then, when you are a company that is doing breeding, that's a very valuable information, right? So that was another part.